Welcome to the channel. Today we are gonna discuss about genetic mutations. So the first thing that comes in our mind, what is mutation? Mutation in molecular terminology is referred to as sudden and heritable changes in genetic sequence of nucleotide bases. It may be permanent or not, and it shows a phenotypic changes. We all know mutations have deleterious effects, and most of the mutations are destructive in nature. Very rarely mutations are advantageous. Mutations like induced mutations, which are used in crop production and in incompatibility issues and in RDT. But most of the mutations are bad for nature. Genetic mutations are responsible for variation in species, which itself triggers the evolution. We all know about the importance of evolution in natural selection, right? Mutations may be permanent or not. It depends upon the direction of mutation. Mutation is sometimes a reversible process. Direction of mutation. When the DNA sequence is unaltered, that means it is in its original form, we call it wild type sequence or allele. And after the mutation, it becomes mutant type sequence. And sometimes I'm calling it's not permanent because sometimes the mutant type goes back to its old form, that is the wild type sequence. It is called reverse mutation, or you can say backward mutation, as it's going backward. And from the wild type sequence to mutant type sequence is called forward mutation. Right? Some mutations are spontaneous in nature. That means the mutation undergoes spontaneously. It's very rare in nature, but it occurs. We call it spontaneous mutation. Spontaneous mutation. And mutation can also be occur by the induction of any kind of mutagenic agents. So it's called induced mutation. First we will discuss about spontaneous mutation. Most of the spontaneous mutations occur due to the replicational errors and transcriptional errors. We all have the clear idea about central dogma. The RNA, to be more precise, mRNA, is formed from DNA via transcription and from mRNA to protein production is done by the translation process. And when there is some defects in DNA level, it goes on after the transcriptional, it forms mRNAs, and the error continues to go, and it undergoes translation, and there forms the defective proteins or wrong proteins. So this type of mutation is called spontaneous mutation. and the second one is induced mutation. We will discuss it in our next video with proper detail. Induced mutations are the results of using of mutagenic agents. Thank you very much for watching. Please subscribe and hit the bell button and share. Let the others know about it, and stay tuned for next time. See you.